Okay, so we are going to crack on with Key Area 3. This area's got some really interesting things. We're actually going to go basically from how we get a photon of light to an image in your brain in this area, or at least the very start of that process. It is going to focus mainly on the membrane structure and the proteins that are embedded in the membrane. Your integral proteins and your peripheral proteins will come to play in this, and we will be talking in this one, in this little lesson, specifically about a couple of transmembrane proteins that we will go into in a little bit more detail. So what we're going to do first is we're actually going to take a look at the whole cell, then focus in on the membrane structure, and whatever's on the sheets of paper, that should make your notes up for this lesson. So our first little bit is our more comprehensive cell structure. There's probably going to be a few things here that you haven't seen before. I'm going to go right from our DNA and our information to make our proteins, right the way up to our protein that's had its post-translational modification and it is ready for doing a task somewhere outside of the cell, and it's been packaged in a nice little exosome, also known as a vesicle, and it will be moving around the body to do something. Anything that's outside the cell we will just consider as this extracellular matrix, which is actually a really interesting fibrous gelatin-like substance that really makes up the mainstay of the space between cells. So in our nucleus we've got our DNA. That DNA will be transcribed into RNA. That RNA will be then translated into a protein, usually not in the cytoplasm, or the cytosol as we will come to call it, at the edge of the rough endoplasmic reticulum. And what's going to happen is those little dots there, they are simply ribosomes, and the protein will be manufactured into the endoplasmic reticulum. That protein will then be passed onto the Golgi body, which will package that protein in a vesicle. It will also do some post-translational modifications. That's that processing bit. Those vesicles are membrane-bound bubbles in the cell. So if I was to draw a general vesicle, and vesicle's basically the family name for any of these membrane-bound spheres that we see in a cell. So we've got our phospholipid bilayer, and in here we might have a protein, we might have something else. It could be ions that we've taken in from outside by pinocytosis, which is something that you would do just above advanced higher. And we've got anything coming into the cell is a process of endocytosis. So you'll get the formation of this vesicle with something packaged inside it, and it'll be coming from the extracellular matrix into the cell. That's endocytosis, and you'll get an endosome. And if you've got exocytosis, you're basically taking something that was inside the cell and you're plopping it outside the cell, wrapped in a cell, in a bilayer, in a membrane. All of these, the endosome, the exosome, and the lysosome, can be referred to as vesicles. You will probably see lysosome sitting as a name by itself. These two at advanced higher are probably just going to be referred to as vesicles. The lysosome has digestive enzymes. They're pretty important for the breakdown of proteins back into amino acids, so you can build up some new proteins again. The Golgi is probably something that we haven't really spoken about before. It's actually a really interesting organelle in our cells, but we're not really going to talk too much about it. You just need to know that it's used for post-translational modifications to proteins, and that Golgi will package those proteins in a bilayer, and that vesicle will possibly undergo exocytosis, and that protein will go and do a job outside of the cell. Sometimes they're actually packaged into vesicles that just stay within the cell. They don't all undergo exocytosis, but really that pathway is what they're wanting you to be able to describe. So I would focus on being able to go from this idea of the nucleus to the rough endoplasmic reticulum, where your protein's formed, your protein that will form in the lumen, so the space inside the rough endoplasmic reticulum is the lumen, and that protein will go from here and it will go into the Golgi. It'll be processed and packaged, and then it'll go out for exocytosis. We do have this smooth endoplasmic reticulum. This is bits of the endoplasmic reticulum that do not have any ribosomes attached, and that is usually specializing in the synthesis of lipids, anything that's got a fat molecule associated with it. Phospholipids, steroids, all associated with fat molecules, and depending on the type of cell, you might have barely any smooth endoplasmic reticulum. However, if you've got a cell that's producing lots of hormones, then you're going to have a really big endoplasmic reticulum, smooth endoplasmic reticulum. So if you are maybe a cell in the pancreas and you're excreting a lot of proteins and hormones, you've probably got a pretty big endoplasmic reticulum. I think the testes and the ovaries also, because they produce a lot of hormones, they're all fat based. They have large smooth endoplasmic reticulums as well. That is not required knowledge. What is required is what's on this sheet though. So what I'd like you to do is, when you've copied it, you probably need to be able to produce this page from memory. So once you've copied it once, I would like you to have a second go at it and see how much of it you can draw of the cell, naming it, what's what, and thinking about it as you can. So that's our cell structure. Just a reminder, and we need to know this stuff going forward. What we're going to focus on is this membrane here at the moment, and we're going to go on and speak about that membrane structure. So, as you are already aware, the lipid membrane is not just phospholipids, it's got proteins embedded in there, and we've got this fluid mosaic model. So what that essentially means with the fluid mosaic model is these proteins are not stationary, they'll be drifting around, and these phospholipids will be drifting too. So it's a very dynamic environment, and it's a dynamic space. Now, when it comes to talking about these membrane-bound proteins, all of these are integral proteins, and they're all transmembrane proteins. So they are connecting that extracellular matrix to the cytosol. There is some transfer of information going on. And if we look at the first one, that's a basic channel protein. Aquaporin is the classic one, basically allows the free movement of water molecules. Interestingly, aquaporins technically, especially in eukaryotes, it's often gated, but we'll just use it as a basic channel protein. Not many of them aren't gated, to be honest, but there are some that are just open little pores that we've got in the cell membrane. So a channel protein would just be one that holds a door open that things can move back and forth from. there are molecules that don't need that channel protein to move through. So molecules like oxygen and carbon dioxide, because they lack any formal charge and they're very, very small, they can actually move through the membrane without too much difficulty. The sodium and potassium, even though technically they are smaller molecules, because they carry a charge, they find it really hard to get through the membrane, so they won't move through the membrane with any degree of ease. And that brings me on to our ion pump. So our ion pump and our ion gated channel are two similar but different things. Now, the ion pump will pump ions against the concentration gradient. So this side has got a high Na positive concentration. Oh, I'm struggling to fit that in. Has a high sodium concentration, and we're actually pumping more sodium into that high concentration, so we're going against the concentration gradient. On this side, we'd actually have a high potassium concentration, and we'd be dragging more potassium into the cell. Now, the energy for this active transport comes from the hydrolysis of ATP. Now, that just means ATP is broken up by a water molecule, and that phosphate will probably attach onto our protein somewhere, cause a conformation change, and that will spew our sodium out to this side here, and it will bring our potassium into this side here. So just to know that the energy to move those molecules comes from the hydrolysis of ATP, and the conformation change, so the shape change in the protein, will allow that process to take place. Now, the ion gated channels work by diffusion, however they're not always open. So what I'm going to do is I'm going to take the example of the potassium here. So we've got a high concentration of potassium, and the potassium's constantly wanting to flow this way, but because this is closed, it can't do that. However, if the cell decides, for whatever reason, it might be a cell in your heart and it's needing to make your heart beat, and so it's needing to create a voltage, it might want to open that and allow the K positive ions to rush through here, and that causes a big change in the charge between the inside of the cell and the outside of the cell, and that can be used for cell signaling. So the opening of those ion channels is really important, and they're gated because they can be either closed or open. Now, there's really two different ways that we can do that. The conformation change to go from closed to open can be either caused by a ligand binding to it, so essentially just some other molecule binding to it, that causes it to open, or it could cause it to close. You could also get a voltage change, so especially when we look at nerve cells, and we're going to look at this in a lot more detail, we could get a voltage change, and that could trigger the opening or closing of these gated ion channels. So those are really the three that we want to concentrate on, and we will speak about them, and we will do a couple of questions on them a little bit later on. I am going to try and embed a couple more questions, but this is really just a start off lesson. Wednesday we're going to have a really big one when we take a look at signaling, which will really bring a lot of this together as well. Now, the last thing that we've got to talk about is facilitated transport. This is basically where we have a molecule that will diffuse, but we're going to give it a little bit of a helping hand. So glucose moving through the glucose symport protein will be assisted by sodium ions and ATP kinase, or KATPase. So it's gonna be passive plus a little bit of active. So it's going to be those things combined, and that can really allow you to take in molecules at a very fast rate. It's also partially because glucose is relatively big. It's not a very small molecule. You've got C6H12O6. So compared to a water molecule in the aquaporin channel, the glucose molecule is really big, and it just needs a little bit of extra help. That extra little bit of help comes from the combination of sodium ions and the KATPase, or it's probably a protein kinase. So the energy is coming from ATP there, and that'll be the hydrolysis of ATP assists with the passive transport of glucose. So that is really it for today. I'm gonna do a couple of little pi mole things to show you what these molecules look like, maybe go over some of the stuff from Key Area 2, and that'll be us. So to start this section off, I just want to go over the last little bit that we did, which was the glucose symport protein. Now that is a gated ion channel. It'll be the conformation change, so the shape change for the protein itself will come from the hydrolysis of ATP and the phosphorylation of that protein, and that'll alter the shape. Now, what's going to happen in terms of the sodium and the glucose is both the sodium and the glucose are in a high concentration outside the cell. So the glucose is going to flood in, but the sodium's going to flood in at the same time. And the glucose is essentially going to get caught up in the current of the sodium. So all of that sodium moving in will help drag the glucose in. I hope that makes that a little bit clearer. You might want to add that little bit into your notes. We can have a bit more of a chat about it. We'll certainly try and find some questions that are related to that to just give it a little bit of clarification. Now, if we switch to our pi mole, we've got, we'll just quickly run through what's going on with these. So here I've got two aquaporin channels. These are, I think the word would be dimerized, because there's two of them and they are the same. So there's two that are cross-linked. I'm not actually sure what those molecules are. Let's find out. Right, what have I just clicked on there? Oh, here we are. BGL. I think that might be a steroid. I think that might be cholesterol. Hold on. BGL protein bank. Oh, that looks like it. It is B2 oxytyl glucoside. No idea what that is. Ladies and gentlemen, this is what we call a rabbit hole.
Wikipedia page. Cool. So it is a non-toxic surfactant. So just for those of you who don't do chemistry, surfactants break up fatty deposits. So, well, once again, I might as well show you. Non-ionic surfactants. Surfactants are compounds that lower the surface tension between two liquids. So they will break up fat so that they can essentially mix with water. Emulsifiers are surfactants. This one's a biological one, so it's not there to break the membrane down entirely, but to make it more soluble. And that probably helps stabilize. So if we take a look at that molecule there is helping stabilize that protein within the membrane. So remember, you'll have membrane all around here. You'll have your phospholipid bilayer, right, really from here to here will be phospholipid bilayer just about. Well, maybe not that much actually. These are probably sticking out quite high from the membrane structure. But you'll have a fairly decent thickness here that will be that membrane structure, and this will be a transmembrane protein. So if you imagine the membrane sticking out both sides, this is the one end sticking out of the cell, this side's sticking into the cell, and we've got some interesting little things hanging about, but we don't want to go into that too much. Just from the last unit, this is mainly made up of alpha helixes, and those little bits there are turns. So we've got lots of alpha helixes and turns for our aquaporin, and we've got this thing down here, which is P. I don't think it's going to be very easy to see. I don't know what that is either. But once again, if you wanted to take this further and you wanted to find out what this little thing is in here, all you would do is go to protein data bank. We'll just type it in here, see if we can search from it. Ooh, I wonder if there was some. Cut ligand. Okay, they all seem like they're the same thing. That seems more like it. Ooh. 1,2-dioyl sin glycerol 3-phosphoethanolamine. Ugh. Well, certainly looks quite long. Okay, we won't go down this rabbit hole. But you can see where, if your interest takes you, you really can find out a lot about this. I would suggest looking at aquaporin, especially if you've got some time in your hands. It's an interesting little protein that facilitates the movement of water between cells. They are, in eukaryotes, they are usually gated, but they can just be channel openings as well. Right, last but not least, we have our glucose symport transmembrane protein, and that is a crazy looking thing. The purple bits are your sodium ions that seem to be embedded. We've also got these, which are ER3. Once again, we could go down a rabbit hole. Now, this is, as you will be aware, those are your alpha helixes. They take up that conformation because of hydrogen bonding. You've got your turns here, which are basically the bits that aren't beta pleated sheets or alpha helixes. They are connecting those major motifs together. And if I very quickly put a surface, oh no, I'll select the whole thing, all, I'll give this a surface. Load. Woo! There we go. That's what it would look like if you were the size of an atom. It would look quite big, but it is very, very small. And this would be a transmembrane protein. It's got a very odd shape, but that is our glucose symport protein. So once again, you can always take the advanced hire really is about you guys learning to take control of what you learn about, and if you want to go down a rabbit hole, it will actually help you. It's the first time, it's really the first SQA course that genuinely going down a little bit of a rabbit hole and learning some new stuff will help you out. It will build your understanding more fully as you go forward. But today, just the notes from the sheets of paper, and have a wee think about how you're going to be able to memorize all of that. Rewriting the cell structure, maybe expanding it out into the membrane would be a good idea. We'll talk maybe a little bit about doing a poster on that later in the week. I do have a really, Wednesday is going to be the big one. We're going to do cell signaling and how basically we amplify very small signals into very big signals, which is quite a cool little bit. But I will leave it there for now, guys, and I hope you're all doing well.